All right, hello everybody. It's 11 o'clock here in Minnesota, so we're going to go ahead and get started. Thanks everyone for joining our webinar this morning. I'm Jackie Carville, the Marketing Director here at ImmunoChemistry Technologies, and I will be moderating this webinar today. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. Simply type your question into the chat dialog at the right and select Send to Host. If you have any technical difficulties during the webinar, you can feel free to send your chat to me, and I'll do my best to get you up and running. We also will be posting the video recording of this webinar either later this afternoon or tomorrow morning, so look out for an email from me after the webinar with a link to this recording. And finally, we are offering a special discount available only to webinar attendees at the end of the presentation today, so stay tuned through the end for that special discount code. Now I'd like to go ahead and introduce our speaker, Dr. Christy Strandberg. Christy is a staff scientist here at ICT and focuses on new product R&D as well as technical support and assay development consultation for our customers. Today she'll be going over some of the products in our oxidative stress product line, including our brand new Total ROS kit. And with that, I'm going to go ahead and turn it over to Christy.
Well, thanks, Jackie. And thank you to all the listeners who are tuning in today. Before I dive into today's webinar topic, I would like to take a quick moment to tell our listeners a little bit about ImmunoChemistry Technologies. We are a privately owned company that was incorporated back in 1994. We began as a company that provided custom assay services. Now, this includes activities like immunoassay design and development, antibody purification and conjugation, and so on. We continue to offer custom assay services. A few years ago, we expanded our company to offer lines of consumable reagents. This includes things like ELISA solutions, such as coating buffers, blockers, diluents, conjugate stabilizers, and such. We also offer a line of products tailored for detecting cell health status. Included in this is ICT's line of products for detecting oxidative stress. So I want to begin today's talk by discussing some of what I consider to be important background information. The first concept I want to introduce is the the oxidation reduction reaction, which is otherwise known as a redox reaction. This is a type of chemical reaction that involves the transfer of electrons between two species. During a redox reaction, the substance, be that a molecule, atom, or ion, changes by either losing an electron, in which case the molecule has been oxidized, or by gaining an electron, which has the end effect of reducing the amount of positive charge, in which case the molecule is said to have been reduced. Now, disturbances in the normal redox state of cells can lead to the buildup of toxic molecules called reactive oxygen species. These are molecules that are capable of damaging DNA, lipids, and proteins. Reactive oxygen species are chemically reactive molecules containing oxygen, as their name implies. From a biological perspective, reactive oxygen and nitrogen species are formed as a natural byproduct of the normal metabolism of oxygen. One such example of ROS production takes place in the mitochondria, which are the organelles responsible for generation of most of a cell's energy. The mitochondria produce the reactive oxygen species called superoxide as a byproduct of normal activity. The superoxide that leaks out from the mitochondria is converted into hydrogen peroxide. Under normal conditions, the detrimental effects of hydrogen peroxide and superoxide are ameliorated by enzymes such as catalase and superoxide dismutase, which break down these damaging molecules into oxygen and water. ROS play important roles in cell signaling and homeostasis, and their production can increase dramatically during times of environmental stress. These molecules also possess antimicrobial properties, and they're involved in the immune response against invading pathogens. Some examples of reactive oxygen species include superoxide and hydrogen peroxide, as well as hydroperoxide, hydroxyl radicals, and peroxynitrite, just to name a few. Now, this slide depicts the formation and decomposition of ROS. I've included this slide just to really give our listeners a better appreciation for the complexity of cellular redox reactions. I won't go into much detail here. I just really want to share this figure to illustrate how complex this system is and to highlight that there are many different enzymes and molecules involved in the formation and decomposition of reactive oxygen species. One example, if you look near the top of this figure, the reduction of molecular oxygen produces superoxide. and actually superoxide serves as the precursor to almost all the other reactive oxygen species. This mutation of superoxide produces hydrogen peroxide. Hydrogen peroxide, in turn, may be partially reduced to form a hydroxyl radical or fully reduced to form water. So under normal physiological conditions, cells control ROS levels by balancing the generation of ROS with their elimination by scavenging molecules such as antioxidants. However, under conditions of oxidative stress, excessive ROS can build up and damage various cellular components, such as DNA, lipids, and proteins. Some of the most harmful effects of reactive oxygen species exposure are the damage of DNA, the oxidation of polyunsaturated fatty acids and lipids, which is also known as lipid peroxidation, oxidation of the amino acids found in proteins, and the oxidative deactivation of specific enzymes. So as I just mentioned, the buildup of ROS can have numerous effects. One such effect is that ultimately oxidative stress can trigger cell death. Moderate levels of oxidative stress can cause a cell to initiate apoptosis, which is a programmed form of cell death. Severe levels of oxidative stress can cause a cell to trigger necrosis. Now, apoptosis occurs through two different pathways, the intrinsic pathway and the extrinsic pathway, which are depicted in the figure shown on this slide. If you would like to learn more about cell death and apoptosis, as well as our products for detecting cell death, please visit our website at www.immunochemistry.com. I've also given a couple webinars in the past where I go into greater detail on apoptosis and necrosis. If you're interested in learning more about these processes, you can access these resources from our webinar library. So an imbalance in the production of reactive oxygen and nitrogen species is associated with many different diseases or pathologies. An overproduction of free radicals can result in the damage of proteins, DNA, and lipids, as I mentioned. The simple cartoon that's shown on this slide portrays lipid peroxidation. The overproduction of ROS has been associated with diseases such as atherosclerosis, diabetes, cardiovascular disease, cancer, and chronic inflammation. Now, the link between cancer and chronic inflammation is actually much more complex than it may appear on the surface. For example, chronic inflammation, which is partially regulated by ROS, is considered a major mediator of cancer. However, modest levels of ROS are required for cancer cells to survive. Although, it should also be noted, excessive levels of ROS can kill cancer cells. So really the take-home message here is that the overproduction of ROS is involved in many disease states. However, the production and elimination of ROS in biological systems can be complex, to say the least. Much of this presentation thus far has focused on the damaging effects caused by the production of reactive oxygen and nitrogen species, but it's important to remember that these molecules serve beneficial functions within the organism as well. In mammals, ROS are induced as an antimicrobial defense during infection. Exposure to ROS can be detrimental to the invading pathogen by causing DNA damage, which ultimately can kill that invading microbe. The importance of such a defense is evident in individuals who have deficiencies in generating ROS. One such example is chronic granulomatous disease. These individuals are highly susceptible to infections caused by a broad range of microorganisms, such as Salmonella enterica, Staphylococcus aureus, Serratia marcescens, and Aspergillus. So, switching gears, I would like to begin introducing ICT's variety of products for detecting oxidative stress. The newest addition to our catalog is the Intracellular Total ROS Activity Assay Kit. The Total ROS Kit utilizes a proprietary oxidation-sensitive probe called Total ROS Green. On its own, this cell-permeant dye is non-fluorescent, but when it's in the presence of intracellular reactive oxygen species, the dye becomes oxidized to its green fluorescence-capable form, making it easy to detect by common methods such as flow cytometry. I'm actually showing you some flow cytometry results on this slide here. The histograms shown here depict the results from a jerk cat suspension cell experiment. These cells were preloaded with Total ROS Green, and then were exposed to 100 micromolar tert-butyl hydroperoxide for 30 minutes to induce the production of reactive oxygen species. The increase in ROS activity is evident in the overlay histogram, which is shown at the far right. The mock-treated cells are represented by the black line, and the tert-butyl hydroperoxide-treated cells are represented by the red line. When you look at this overlay, you can see the nice shift in median fluorescence intensity in the cells that were exposed to the ROS-inducing reagent. Hydrogen peroxide is a molecule of great interest as well. In biological systems, the one electron or incomplete reduction of molecular oxygen that occurs during respiration produces the highly reactive superoxide molecule I mentioned before. And as I also mentioned, superoxide it serves as a source for most other reactive oxygen species. So reduction of superoxide by superoxide dismutase results in the production of hydrogen peroxide. Hydrogen peroxide is unique in that it's a relatively stable oxygen form, which unlike it's a more oxidized precursor, it's capable of readily penetrating cell membrane barriers and influencing intracellular metabolic activities. Now, hydrogen peroxide has important roles as a signaling molecule in a variety of biological processes. For example, hydrogen peroxide is thought to serve as a signal to immune cells to converge on a site and initiate defense and promote healing. ICT offers both a fluorescent and a colorimetric hydrogen peroxide detection kit. These kits are available in a 96-well format and are designed to quantitate hydrogen peroxide in a variety of samples, such as fresh urine, buffers, and tissue culture medium. The graph shown here depicts a typical standard curve from our hydrogen peroxide fluorescence detection kit. Now, in this particular kit, we include a hydrogen peroxide standard that you use to generate your standard curve. Samples are mixed with a fluorescent substrate, and the reaction is then initiated by the addition of horseradish peroxidase. Samples are then incubated at room temperature for about 15 minutes, and any hydrogen peroxide present in the sample will oxidize that horseradish peroxidase, which in turn oxidizes the colorless non-fluorescent substrate. This converts it into a fluorescence-capable product. What you have in the end is that if you have increasing concentrations of hydrogen peroxide in your sample, this corresponds to an increase in the amount of signal of our fluorescent product. So that is how overview of how this little kit works. As I mentioned before, DNA is one of the main molecules that can be damaged during times of oxidative stress. Hydroxylation of the guanosine found in DNA occurs in response to both normal metabolic processes and in response to a variety of environmental factors, i.e. anything that increases reactive oxygen and nitrogen species. Hydroxylation of guanosine is depicted in the diagram shown to the right. So 8-hydroxy-2-deoxyguanosine, or as we'll commonly refer to it as 8-OHDG, is produced by the oxidative damage of DNA by reactive oxygen and nitrogen species. And really, this is a pretty well-known, well-studied marker. This is considered to be a well-established marker for oxidative stress. Increased levels of 8-OHDG are associated with the aging process, as well as a number of pathological conditions, including cancer, diabetes, and hypertension. Oxidation of DNA can cause many types of DNA damage, including single-strand breaks, protein DNA adducts, as well as intra- and interstrand crosslinks. ICT offers a competitive ELISA for the quantitation of 8-hydroxy-2-deoxyguanosine. Our kit's compatible with a variety of sample types, including urine, cell culture media, plasma, tissue samples, and saliva. Now, this assay utilizes an 8-hydroxy-2-deoxyguanosine-coated plate that has been paired with an HRP-conjugated detection antibody that's specific for 8-OHDG. Now, there's a typical standard curve that's shown in the graph on this slide. As this is a competitive ELISA, the greater the concentration of 8-hydroxy-2-deoxyguanosine in the sample, the lower the end signal. So how this works is that the greater the amount of 8-OHDG in your unknown sample, the more likely it is that the detection antibody will bind to that free 8-OHDG present in the sample instead of binding to the 8-OHDG that's been coated onto the plate. And only the detection antibody that is bound to coated 8-OHDG will be retained and therefore detected. Antibody that binds to the free 8-OHDG will be washed away. So that's how you end up with a lower signal for the higher concentration. Now, the oxidative stress can also affect the mitochondria. Mitochondria are double membrane-bound eukaryotic organelle that are found in most cell types. The inner membrane of the mitochondria is where cellular respiration takes place. This process is responsible for generating most of a cell's energy in the form of adenosine triphosphate, or ATP. Mitochondria produce energy by establishing an electrochemical proton motive force across their inner cell membrane. This fuels synthesis of ATP. The electron transport chain is involved here and is actually responsible for creating this electrochemical proton gradient. This slide shows how mitochondria produce energy by establishing this electrochemical proton motive force across their inner cell membrane. As I mentioned, the electron transport chain is responsible for creating this electrochemical proton gradient. Now, ICT offers several potentiometric dyes that can be used to assess the status of this proton gradient. These dyes are called TMRE, TMRM, and JC1. The image shown here on this slide depicts the accumulation of TMRM. Inside a healthy cell, this positively charged lipophilic dye enters the negatively charged mitochondria, where it accumulates and then fluoresces upon excitation. Oxidative stress can trigger the depolarization of the inner mitochondrial membrane, which results in a dissipation of the protons that had accumulated there within that intermembrane space. Depolarized or inactive mitochondria have decreased membrane potential and fail to sequester dyes such as TMRM, as well as TMRE and JC1. The end result here is that you have a measurable drop in the fluorescence signal, which can be easily measured by flow cytometry and fluorescence microscopy, etc. To show you some sample data depicting this, here's some results of studies we've done in-house that show the measurable drop in fluorescence signal as mitochondrial inner membranes depolarize. Here we're looking at some sample flow cytometry and microscopy results. Both of these experiments used Jurkat cells that were treated with 1 micromolar staurosporin for 3 hours to induce apoptosis, and were then stained with either TMRM or TMRE. The staurosporin-treated samples are shown as the red peak on the flow histogram and the topmost microscopy images. Samples were exposed to DMSO as a negative control and then stained with either TMRM or TMRE. So if you look at the flow cytometry results shown up top, the white peak that's outlined with a black line on the flow histogram, that's the untreated or mock-treated DMSO negative control cells. Oh, sorry, I said that wrong. The red line is the mock-treated cells, and the black line represents the cells that were exposed to staurosporin. So there's a loss in fluorescence signal when the cells depolarize their inner mitochondrial membranes. If you look at the fluorescence microscopy results shown below, you can see the negative control cells on top, and they're staining more brightly red than the cells that were treated with staurosporin below. Now, I'd like to switch gears slightly and discuss ICT's products for detecting nitric oxide and nitrosative stress. Nitric oxide is an important physiological regulator of systems such as the cardiovascular system, the nervous system, and the immune system. Nitric oxide is made bioavailable by 2 routes. It can be generated through the activity of the enzyme called nitric oxide synthase, or it can be ingested in the form of nitrates and nitrites and then rapidly converted into nitric oxide. Nitric oxide is known to have cytotoxic effects, for example, when released by macrophages during an immune response. Additionally, because nitric oxide is highly reactive, it can be converted into a toxic radical responsible for causing nitrosative stress. Now, ICT offers a colorimetric kit for detecting both endogenous nitrite as well as total nitric oxide. This kit has 2 assay capabilities. The first assay is designed to quantitatively measure endogenous nitrite. During the second assay, nitrate is first converted to nitrite using nitrate reductase, and then is quantified as well, resulting in a measurement that correlates the amount of total nitric oxide. The nitrate concentration can be obtained by then subtracting the endogenous nitrite result from the total nitric oxide result. This kit has been designed to be compatible with water and buffers, as well as serum and plasma samples, also compatible with urine, saliva, and tissue samples. And the data that I'm showing depicted on this slide represents a typical standard curve generated from using this well-based kit. ICT is also preparing to launch a new oxidative stress kit designed for the detection of intracellular nitric oxide synthase activity. This kit utilizes a non-fluorescent cell permeant reagent called diaminofluorescein 2-diacetate, or DAF2-DA. DAF2-DA is used to detect free nitric oxide and nitric oxide synthase activity in living cells. Once DAF2-DA is inside the cell, cytosolic esterases remove the diacetate groups, releasing DAF2 and thus sequestering this reagent inside the cell. Production of nitric oxide converts this non-fluorescent dye to its fluorescent triazole derivative, which can be observed by flow cytometry, fluorescent plate reader, or by fluorescent microscope. This slide shows fluorescence plate reader and flow cytometry results from experiments where Jurkat cells were preloaded with DAF2-DA and then were either mock treated or were exposed to 1 millimolar diethylamine nanoate. They were exposed to this for about 30 minutes to induce NOS activity. Now, if you look at the plate reader results, which are shown in the bar graph on the left, you can see a significant increase in the relative fluorescent units when comparing the induced cells with the mock-treated cells. The slide also shows flow cytometry results for induced and mock-treated Jurkat cells. Here, again, you can see a marked shift in the mean fluorescence intensity when comparing the mock-treated cells from the top histogram with the induced cells shown in the lower histogram. Approximately 95.5% of the induced cells stained positive, compared to the mock-treated cells where only about 3.3% of the population stained positive. So I want to switch gears again somewhat to mention some of our products that are geared more towards detecting the mechanisms for controlling oxidative stress. So as I mentioned earlier in this talk, cells use molecules such as antioxidants to help repair the damage caused by oxidative stress. Examples of such molecules include superoxide dismutase, catalase, and glutathione peroxidase. Glutathione, or GSH, is involved in the regulation of many essential biochemical processes, including DNA synthesis and cell proliferation. Intracellular levels of glutathione can favor a cell death pathway via apoptosis. This occurs when there's adequate stores of intracellular glutathione. When glutathione stores have been depleted, cells tend to die via necrosis or autophagy. Harmful cellular levels of hydrogen peroxide are minimized by the enzyme glutathione peroxidase. This happens in a manner that utilizes glutathione as a reductant. The lower image shown on this slide depicts the oxidation of a glutathione dimer, or GSSG, as the glutathione peroxidase converts hydrogen peroxide into 2 molecules of water. Glutathione reductase converts the glutathione dimer back into 2 glutathione molecules. So ICT's glutathione kits are designed to detect and quantitate both free and oxidized glutathione. Our kits have been validated to work with many sample types, including whole human blood as well as isolated erythrocytes, serum, plasma, and urine. Our kits are also compatible with most cell lysates and tissue homogenates. The kit shown here utilizes a proprietary molecule that will covalently bind to a free thiol group on glutathione to yield a highly fluorescent or colored product. Free glutathione is measured first, followed by the addition of a molecule that converts the oxidized glutathione into free glutathione. Then a second measurement is taken, allowing the end user to calculate the total amount of both free and oxidized glutathione present in their unknown sample. The data shown here represents a typical standard curve generated using the fluorescent glutathione detection kit. I want to point out ICT also sells a colorimetric version of this kit. For more details on those, please visit our website. ICT also offers a kit for detecting intracellular levels of glutathione. Our intracellular GSH kit provides all of the essential reagents for monitoring changes in intracellular glutathione levels by flow cytometry. The key reagent in this assay is what we call the thiol bright green dye. This proprietary thiol reactive dye quickly penetrates cell membrane structures and accumulates primarily within the cytosol of living cells. In the presence of free thiol-containing molecules such as glutathione, the non-fluorescent thiol bright dye covalently binds to glutathione. This process converts the dye to its green fluorescence-capable form. During periods of oxidative stress or glutathione depletion associated with cell death processes such as apoptosis, the cytosolic concentrations of the green fluorescence dye become significantly diminished. The reduction in intracellular glutathione concentration directly translates into an easily monitored reduction in green fluorescence output within the dying or oxidatively stressed cell population. So I'm showing you on this slide are some sample flow cytometry results where Jurkat cells were either treated with DMSO as a negative control or were exposed to 1 micromolar staurosporin to induce apoptosis. The effects of staurosporin on the level of intracellular glutathione can be seen in the overlay histograph. The green line represents glutathione levels in mock-treated cells, and the red line represents the diminished glutathione level in the apoptotic cells. So that wraps up our panel of oxidative stress products. I do want to take a quick moment to recap the products I've introduced here. ICT carries products for detecting intracellular ROS activity, hydrogen peroxide, DNA damage in the form of 8-hydroxy-2-deoxyguanosine, mitochondrial membrane status, nitric oxide synthase, and glutathione. I would also like to remind our listeners that ICT offers a variety of other fluorescent probes. In addition to the products covered in today's webinar, we also carry a number of products for the detection of caspases, cathepsins, and serine proteases, as well as other auxiliary stains like propydium iodide and Hooks. If you have any questions about other products or services offered by ICT, please contact us by emailing help@immunochemistry.com or by calling us at 1-800-829-3194. In case it's of any help, here is our contact information. And then lastly, I want to put up that promotion that Jackie had mentioned at the start of this webinar. So if anyone's interested in our the 9137 or 9144 kits, these are the total ROS kits or the intracellular glutathione detection kits. We're currently offering a 10% off plus free shipping promotion for those two products. And with that, I guess I'd like to open up for any questions we might have.
Wonderful. Thank you so much, Christy. We're having lots of questions chatted in by people, so feel free to keep chatting those in. In case we don't get to your question or your question requires a little bit of a more in-depth technical response, we will follow up with everybody's questions personally after the webinar. So, if we don't get to you right now, don't worry, we will get back to you. But I'll start kind of going through and taking some of the questions that have been chatted in. Someone's wondering, what is the most reactive form of all ROS?
Oh, great question. So I think the most reactive form of reactive oxygen species would be the hydroxyl radical. This one has a single unpaired electron, so it can react with oxygen in a triple ground state. The hydroxyl radical interacts with all biological molecules, and it can cause subsequent cellular damage such as lipid peroxidation, protein damage, and membrane destruction. Really, I would say this is the most damaging because cells have no enzymatic mechanism to eliminate this form of reactive oxygen species, so its excessive production can eventually lead to cell death.
Wonderful. Thank you, Christy. We also have someone else wondering, can they directly assay whole living cell suspensions for changes in intracellular hydrogen peroxide levels with our hydrogen peroxide detection kit?
Well, no. Unfortunately, this fluorescence-based hydrogen peroxide detection kit requires the use of a clarified soluble sample matrix. So that's why we recommend you use this to test samples such as like urine or cell culture media or samples that are in a buffer. Whole cell targets will require some sort of cell lysis step. So we've really not worked with these kits in a whole cell situation.
Gotcha. Thank you, Christy. Another question that's come in, someone's wondering what intracellular sites are known to generate ROS?
Another great question. So different organelles within the cell can generate reactive oxygen species. If you remember from earlier in the webinar, I talked about how mitochondria are a site for ROS production. They produce superoxide. Other cellular locations that are known to have ROS production include the endoplasmic reticulum, particularly during times of endoplasmic reticulum stress, as well as peroxisomes. In the peroxisome, ROS production occurs during the metabolism of long-chain fatty acids. In addition, various enzymes, including oxidases and oxygenases, can generate ROS as a part of their enzymatic reaction cycles.
Great, thank you. We have another question with regards to our various mitochondrial kits. So someone's wondering how the TMRE, TMRM, and JC1 dyes differ from one another.
Sure. So that answer requires a little more detail. From a functional standpoint, JC1, TMRM, and TMRE are all very similar. These are all considered to be cationic lipophilic dyes that accumulate in healthy or polarized mitochondrial membranes. Of these three products, TMRE and TMRM are really closely related and are chemically very similar. TMRE stands for tetramethylrhodamine ethyl ester. TMRM is tetramethylrhodamine methyl ester. So of these two dyes, TMRE is slightly brighter and has a slightly greater fluorescence potential. TMRM exhibits the lowest mitochondrial binding and electron transport chain inhibition of the two dyes, but really these are very, very close. JC1 is a little bit more unique in that it exhibits a dual fluorescence property. In healthy cells with a high mitochondrial membrane potential, JC1 spontaneously forms complexes known as J aggregates that have an intense red fluorescence. On the other hand, in apoptotic or other unhealthy cell situations where there's low mitochondrial membrane potential, JC1 remains in a monomeric form, which shows only green fluorescence. So flow cytometry analysis of JC1 can be performed using two-color red versus green fluorescence plots. Just want to mention this in case any of our listeners are interested in more information on our mitochondrial membrane potential dyes. We will be having an upcoming webinar that focuses exclusively on this topic, so definitely stay tuned. There'll be future announcements when that one's coming out.
Thanks, Christy. So in interest to keep this webinar short for everybody, I think we're just going to take one more question now. Again, if we're unable to get to other questions, feel free to keep chatting them in. We'll follow up with you within the next day or so here with a specific answer for that. But I guess the last question we'll take today, someone's wondering what is the most stable form of ROS?
Yes, if you remember back to earlier in the webinar, I was talking about hydrogen peroxide. This is thought to be the most stable form of reactive oxygen species. Not only is it the most stable form, but I also want to add that it's thought to be able to produce greater effects on cell components compared to other ROS species. This is because in part because of its stability, but also because of its ability to traverse the internal membrane structures. So as I mentioned, hydrogen peroxide is cell permeable. So it can get into different cell compartments and exert more of an effect.
Thanks, Christy. All right. Well, again, that's about all the time we have for today. Again, if we're unable to get to your question, we'll follow up with you personally right here after the webinar. Again, I will also be sending out this webinar recording within the next day or so, so keep an eye out for an email from me with that. As Christy mentioned, our next webinar is scheduled for August 24th and is going to be on the topic of solutions to detect mitochondrial membrane permeability. And this webinar registration is available, so you can go ahead and register for this webinar right now on our webinars page. If you can think of any other questions after the webinar or have ideas for topics for future webinars that you'd like us to cover, you can feel free to fill out the Contact Us form on our website. You can also reach us through social media on Facebook, LinkedIn, or Twitter. And thank you again, everyone, so much for joining us, and have a great rest of your day.